So let's apply what we have just learned to plant cells and animal cells and see if there are any differences in how these different solute concentrations can affect the cell. So on the left-hand side, we have red blood cells. So red blood cells should look like the ones in the middle here under B. So red blood cells are nice and circular, and they have little dimples in the middle. So let's say we have red blood cells in an isotonic solution. So isotonic means equal. So that means the solute concentration inside of the red blood cell is equal to the solute concentration outside of the red blood cell. And this is looking really good here for our red blood cells. All right, so this is the perfect environment for our red blood cells. On the other hand, if we take our red blood cells and we place it in a hypotonic solution, so the outside around the red blood cells has a lesser solute concentration compared to inside of our red blood cells, what we're going to see that's going to happen is that water is going to move from a hypotonic environment to a hypertonic environment. So water is going to move into our red blood cells. And there's nothing to prevent that water from keep on flowing inward because our animal cells are only surrounded by a plasma membrane. And remember, membranes are very flexible. So it can stretch to a certain point, but if the water keeps on coming in, our red blood cells will burst. And obviously, this is not a good condition to be in. So if you think about it, right, when you go to the hospital and they give you an IV, and it could be that you're dehydrated and you just need fluids, why is it that they don't just give you pure water in your IV? Usually IVs are saline solutions. It's like a 0.9% saline or salty solution. So they're giving you salt instead of just water. And the idea is that you need something that is isotonic to your cells. You don't want something that is hypotonic to your cells because it could be deadly to your cells. Here we have a hypertonic environment. So the solution that the red blood cells have been placed in has more solutes than the inside of the red blood cell. So water will move from hypotonic inside the red blood cell to outside, right? So water is exiting our red blood cells. And because of that, they look all shriveled up, right? They've what we've called plasmolyzed, all right? So they're, or crinated, is the term for red blood cells, okay? So they've shriveled up, and this obviously is not good either for the red blood cells. Now, let's cross over to a plant cell. So this cell here has a cell wall around it. So plants have a very rigid cell wall that helps to give them support. And inside here, we see the cytoplasm has pulled inward when a plant cell is placed in a hypertonic environment. So the outside of the cell is hypertonic, having more solutes as compared to inside of the cell. And just like we saw over here with the red blood cells, that means that water is going to exit the cell. So it's going to lose water. And this can be very deadly to a plant cell if it loses too much water. So I always think of when I'm fertilizing my lawn and I've dumped too much fertilizer on the grass in one spot, and when that happens, you get this kind of burnt patch in your lawn because of too much fertilizer. And really what you've done is you've added too much solute to your lawn. And that's created a very hypertonic environment. And because of it, the plants cannot take up water. They actually lose water and they die because they've lost too much water. Now, what if we place a plant in an isotonic environment? This was perfect for our red blood cells, but it's not necessarily perfect for our plant cells. So here in isotonic, we have water going in and we have water going out, but it's just not enough water for a plant cell. So in an isotonic environment, your plants will be wilted. They will fall over. They have water, they're okay, but they're wilted. If we place our plant in a hypotonic environment so that the inside has a greater solute concentration than the outside, water is going to move into our plant cell. This is what we want. Notice the outside of it looks like it's kind of bulging compared to our isotonic picture over here. So all this water moving into our plant cell is putting pressure on the cell wall. Now our plant is standing upright. It's no longer wilting at all. So this is the perfect environment for a plant cell, is a hypotonic environment. So everything that we've been talking about so far has been passive transport. Diffusion, facilitated diffusion, and osmosis are all types of passive transport. Active transport is another means of moving material across the membrane. In this case, though, since it's active, we need energy. And we're going to use energy in the form of what's called ATP a lot of times. And this is kind of a universal energy currency that we use to move materials within a cell. So active transport involves moving against a concentration gradient. So there are times when you might want to stockpile certain molecules within the cell or get rid of something completely from a cell. So you're moving against a gradient. So here we have our transport proteins, and these are required for active transport. We see energy coming in to help this transport protein pump these sodium ions from where they are very low concentration, and we're going to pump them to an area where there's a much higher concentration. So this is active transport. We're moving against the concentration gradient, but in order to do so, we need some energy. All right, we're stockpiling something. We're going the opposite of our usual diffusion. There are other types of movements. These usually involve much larger molecules or materials or organelles or even cells. So there's what's called endocytosis, endo referring to inside, CYT referring to our cell. So this is the process of moving material into the cell, but it's different than what we were talking about with diffusion because we're moving a lot of material in one step. So endocytosis is taking this membrane here, typically the plasma membrane or the outer membrane, of the cell. And you can see these stars and these triangles and these kidney beans on the outside of the cell. And let's say we want to take some of this and pull it inside the cell. We want to get these things across the membrane, but these are way too big to move through a transport protein. So instead, the plasma membrane invaginates, so it just kind of dips towards the inside of the cell, and anything that's out here can kind of flow into that little depression that's made. And then the two ends of the plasma membrane will seal up, right? They will connect together. And once those connect, right, then you have the formation of a vacuole. So this is actually a membrane, right? It's part of the plasma membrane, which is on the outside of the cell. That is now on the inside, but it's completely encircling this kind of droplet of liquid with our kidney bean inside of it. So we took it from the outside and we've pulled it to the inside and formed a membrane around it. So this can form a food vacuole inside the cell. So this is endocytosis, bulk transfer of material into the cell. The opposite is exocytosis. So exo for outward, out of the cell. So in this case, we start, right, with our vesicle or our vacuole that has, let's say, waste material in it. So it's surrounded by a membrane. It is going to travel up to the plasma membrane, and the membrane of the vesicle will fuse with the outer membrane. And when it does that, it's dumping the contents to the outside of the cell. So if you think about it, it's basically exocytosis is just the reverse of endocytosis. All right. So one is pulling membrane in with everything that was outside, gets packaged up in the vacuole. Now we have this membrane-bound vacuole or vesicle with material in it, and we're just going to push it up against the membrane and fuse these two membranes together, and that will open up the contents to the outside of the cell. All right, so question time. Let's see if you can answer a question. Test your knowledge. We have an animal cell that is placed in a hypotonic solution. What will occur? So for these, I always like to kind of draw something out, right? So there's my liquid and there's my animal cell. So an animal cell is placed in a hypotonic solution. We know that hypo means less. So the solution has to be hypo. That means the inside of our cell has to be hyper. I'm going to run out of room to write hyper here. All right. But it's hypertonic. So it has greater solutes compared to the outside. So we know that water will move from hypo to hyper. So water will go into the cell. So which one of that matches this? It says the cell will rupture due to too much water flow coming in. I kind of like that. The cell will shrivel due to too much water loss. Nope, it's not going to shrivel. The cell will remain the same size and be healthy. Nope, water is going to go into the cell. So A is our answer there for that question. Let's try another one. True or false? Large polar molecules and ions are able to move across the cell membrane by simple diffusion. What do you think? So we want to look at the word large polar molecules and ions. Remember, ions have charges. Polar molecules have partial positive or partial negative charges. Are they able to diffuse across the cell membrane by simple diffusion? That's going to be false, right? They need the help of a transport protein. They can move across by facilitated diffusion instead. And then last, let's try this question. Why is cholesterol important for animal cell membranes? Why is cholesterol important in membranes? Is it a transport protein and aids diffusion? Nope. It's not a protein at all. Cholesterol is a lipid. It belongs to the group of lipids. It is used in active transport and passive transport of molecules. Once again, it's not a protein. It's not a transport protein. So C, it keeps the membrane from being too fluid. That might sound kind of good, right? But let's look at D. The molecule is hydrophobic and hydrophilic and will form the phospholipid bilayer. That's actually describing a phospholipid molecule, not cholesterol. So C is our answer. It keeps the membrane from being too fluid. Remember, it keeps the phospholipids from moving too far or flowing too far. So C is our answer.